We'll start out immediately by thanking the sponsors. PCT and GE are sponsoring this workshop. We have a panel of true experts in this field in the CMC side of cell therapy development represented here. And in a moment, I will ask each of the speakers to introduce himself to you, and then we'll move on from there. I'd like to set the stage before we get started, though, and say that the first interactive exercise is that in front of you is a survey, and we're performing a survey to evaluate challenges within CMC to better understand what people see as the main challenges. And I would ask you through the course of this session to check them off. If you have more challenges after the session, then we haven't done our job. So just please fill it in at any opportunity, and then hand it in to one of the staff on the way out, if you would. Thank you very much. So, for those of us who have been at the CMC game in cell therapy, there's no surprise, and this will not come as a shock to you, that changes during process have presented a very unique set of challenges to us. And changes in any manufacturing process are indeed difficult. One of the transitions this industry has been making over the past 30 years or so is an industry that comes from more research-minded to a more really true development and industrialization and commercialization mindset. That mindset brings with it a very different set of challenges. And they're quite incongruent with each other in many ways, in that in the science field and in the side of development, there's more questions to ask, and answers are great, and more questions to ask, and creativity is wonderful. And then as we march towards industrialization, those changes become enemies, and really locking down processes and making them as consistent and robust and high quality, scalable and sustainable, and reduce the cost of goods, and doing all of that which will make them deliverable become real serious challenges for us. Because for a number of reasons, we're not quite ready to make those changes. And why are we not ready? Well, typically we're not ready because when we first started to think about cell therapy, and when cells started to become thought of as a mode of action, there were some very simple cells like red blood cells, and we knew pretty much what red blood cells do. They carry oxygen, so as long as we can maintain that activity and return them, then we should be okay. So it's actually a very simple mode of action, very simple potency, if you will, because there's apparently one mode of action that we can look at. As we've gotten into any more complex cells, anything that has a nucleus particularly, and anything that we're expecting to do something a little bit more physiological, and we're asking these cells to do quite a bit, we did this because we recognized and acknowledged that the current modalities of medicine were missing the point. They would cause remissions in the most case. They had toxicities associated with them, that by and large diseases would return. So these palliative treatments were just not working the way we would like to see them work. So we had these grand ideas. Let's look at cells and see if we can provide for cures. If cells knew what to do in the first place, then they ought to be able to know what to do again. If we take them out of the body, perhaps do something to them, put them back in a different place or in a different way or in a different concentration, and that should work. But that's really abdicating the responsibility of mechanism of action to the cell. And that says we don't really know what to do, so we'll ask the cell to do it. So from a scientific standpoint, it's quite interesting and made for a lot of great research and a lot of great movement. But from a product development standpoint, that presents a challenge, because now all we can really do is propose a mechanism of action. And we can have POMOAs, proposed or purported mechanisms of action, but we really don't know exactly what the cells do. As we're marching down the line, and cell therapy and gene therapy particularly getting much more sophisticated in the ex vivo manipulation of genes, we're learning more and more about how these cells engage at the target and maybe what they do in the first couple of steps, but we still really don't have a complete picture. So that presents an interesting problem for us, because when we make changes to the manufacturing process, which we absolutely do need to make as we're doing development work, the questions that we're asking are, do we change the mechanism of action? Do we change the potency of the cell? Are we altering the cell from doing its primary action, what it is intended to do when it goes back into the patient? And it's a really difficult thing to do. So we develop functional assays so that we can determine at the end of the process whether or not the product that we now have looks enough like the product that we had before that we shouldn't have affected the primary or the proposed mechanism of action. These functional assays have traditionally had issues with them. They're biologic assays, so I don't have to say anything more about that. That's difficulty in itself. That provides some degree of variability. They're new assays. They're difficult to validate because what they're supposed to be measuring is difficult to validate. So in essence, we have proposed functional assays to describe potency that link in some way to a proposed mechanism of action. So then we step back and say, well, we can't do a biologic assay every time we release a product, so we do a snapshot assay, flow cytometry, an ELISpot, something. We look at what the cell produces in terms of cytokines and whatnot, and maybe take other kinds of pictures of cells and look at them as an identity matrix. But we're not really sure how that maps exactly to the functional assay. And again, just to remind you, we're not sure how that functional assay maps to the actual activity of the cell. So we begin to establish, at least on the identity and the potency side, we begin to establish a connect the dots exercise that says if we the cell product kind of looks like this in this range, then it probably should look like that in the activity profile, which should probably be okay. And that argument then is presented to regulators. It builds the spine around which all of our development work is done, and there's discomfort on both sides. And so the challenge that we've had is how do we make sense of that story? And there's a whole set of assays from the safety side as well that's faced similar, but different challenges, but similar to that. And so, you know, this introduction could have been given 10 years ago or 15 years ago. And the question that we're asking today, and we'd like the audience to participate both in terms of questions and answers to this question, is what's happening? How are regulators dealing with this? At the second part of the workshop, Jiwen Zhang from GE will discuss how does this issue map in these cases now, particularly where, as we've been talking about this whole conference now, we're so close to industrialization on a much wider scale. And, you know, has the clinical activity and the clinical transformative nature of these products gotten out ahead of our ability from a CMC perspective to understand exactly what these products do? Because not only will we need to make changes pre-launch and post-launch, but we will need to have multi-site manufacturing. And in multi-site manufacturing, you'll need to have, you could do comparability exercises between different sites. How do we truly measure comparability with a less than perfect understanding of the profile? So there's a number of regulatory strategies that have been taken. Companies have been marching through this. And one of the stated objectives of ARM for the last year and a half now, and strongly going into 2017, will be to provide for assistance and work with regulatory bodies between industry and regulatory and academia to provide for a predictable regulatory framework that is harmonized to the extent we can do that around the world, allowing us to develop our products in such a way that if a regulator says, okay, that checks the box, and at the same time, we know from a biologic perspective that makes sense too. And that marriage and that working together has been going on. There has been a tremendous amount of progress made. There have been products approved. And now we're asking the question, how did people do that? And how can we provide assistance to the rest of the community to help us walk through that? And what's next? So without further ado, I'd like to introduce our panel, and we'll start with Keith Wonnacott. Keith, would you introduce yourself?
Yes, my name is Keith Wonnacott. I work for Novartis Pharmaceuticals. I've been at Novartis for 2 years, and prior to working at Novartis, I was the chief of the cellular therapies branch at the FDA and I was at FDA for 13 years. So that's it.
Francis Meikle. I'm the Director of Cell Therapy Manufacturing at Janssen R&D, Johnson & Johnson. I've been in that area for about 2 years. Before that, I was in antibody development at Johnson & Johnson, and before that in vaccine development at Merck.
Good morning, I'm Stuart Abbott. I'm the Chief Development Officer at Fate Therapeutics here in sunny San Diego. Been with Fate for about 1.5 years. Before that, I was with Celgene Cellular Therapeutics for 8 years. So that gives me about 10 years of history in this field, and I still think I'm a beginner. So we'll see how the panel goes.
Good morning, everyone. I'm Yoshiaki Maruyama from PMDA. I'm now a review director in the Office of Cellular and Tissue-Based Products. I work on 8 years in PMDA, and then currently I is a review director at our office, but before that I work for the Japanese pharmacopoeia. So, thank you.
Hi, I'm Steve O. I'm the chief of the Cell Therapies branch, Office of Cell Tissue and Gene Therapy at FDA. I've been in FDA for about 9, almost 10 years. Our branch, Cell Therapies branch, reviews mostly CMC parts of the submissions, and we have a variety of products that has to do with cell therapies, tissue-derived, tissue-engineered products, including combination products and some of the unique devices.
Hello, good morning. I'm Nicolas Ferry from France. I'm a former scientist and MD, and I've been at the French Regulatory Agency for the past 5 years, where I was in charge of cell and gene therapy. And I was also a member of the CAT, the Committee for Advanced Therapies, at the EMA in London.
So thank you, gentlemen. So I'll start the questions off, but I would just like to point out, if you didn't pick it up, and I'm sure you did, if you're listening at all, what we have here are regulators from around the world, industry professionals who have either been on the regulatory side or not, but in any case have either late-stage products or not, but have been working in this industry for a long time. This presents a very unique opportunity to really dig into some of the solution sets that have been ongoing, to ask the question, how do we navigate within our own territories and outside of them to create global delivery for these products. I'd like to just start out, because in the pre-conversations that we had, there was quite a bit of talk, and Nicholas, I think you started this off, about the differences between gene-modified cells and cell therapy in general, and your thoughts, and perhaps a little bit about the way the regulators in Europe are thinking about the difference in those products, and how this issue of potency and changes around potency and CMC development are affected by the fact that there's another component, a viral component in there typically, or maybe something else, but then also the changes to that cell and how that helps or presents a set of challenges.
Yeah, I think that, as you have said in the introduction, what is really important for us is to know exactly how the product is working, and what is the potency assay that will demonstrate that you know exactly what the cells are doing. And if we look at gene-modified cells, for example, we know that the gene modification will drive the cells to a certain mode of action, and that the action of the cell will be in many, many ways directed by the expression of the gene in the cells. So starting from this knowledge, we can assess whether or not a modification in the process will affect the final biological activity of the cells. Instead, when we are dealing with cells with which we don't know exactly how they are doing, as Bob said, for example, mesenchymal stem cells, which are expected to do very different things in the body, any change in the manufacturing process is difficult to assess, because if you don't know exactly what is in the black box, because cells sometimes can really be seen as black box, what will be the outcome of the impact of the modification on the safety and on the efficacy of the cell is much more difficult to assess. So this is why probably, I mean, when you have a clear modification, such as gene modification, that will really make, in a certain way, more simple the biological assessment of the cells, it's easier for a regulator to know exactly or to understand the impact of the modification.
So that being said, in Europe, there is one product that, in mesenchymal stem cells, are an interesting example of that have been widely analyzed and approved, well, maybe not approved for use, but I guess in Japan, through the conditional approval process, they've been approved. How did you look at this in Japan? How did PMDA evaluate those cells, given that there's no specific gene modification? This is GVHD is one particular therapeutic application. Could you just address that, please?
For that, you are talking about mesenchymal?
Yes, stem cell. Yes.
Stem cell?
Mm-hmm.
Okay. Yes, that's first approved for the autologous product. Allogenic product in Japan, and then so still that mesenchymal stem cell, so that indication is for the acute GVHD, but the potency means potency is multiple. They have lots of biological function in that cell. So we assessed, we reviewed that potency assay done by sponsors. They get many potency assay, but still we actually don't know that which one is a major potency or major to the potency, but still we asking to get the some pharmacological action to still, this is already approved product, but still we asking to get that data as we said the verification. Still we are still discussing about which one is the most effective to that mode of action.
So Yoshiki, that's interesting, but and if this is a question you can't answer, it's okay, but I think I'll try.
Yeah, okay.
If there were no conditional approval mechanism and you were approving this as a licensed product,
mm-hmm. Yeah.
Would that have changed? Would that assessment have changed? I mean, is the reason that we were able to do this is simply because of the conditional approval, and we can work out this later because it's got a very safe profile?
Yeah. So. Actually, Temcell is a full approval product.
Oh, it's a full, I'm sorry.
Yeah, conditional time limited approved is a hot sheet for the autologous myoblastic in the heart failure. So that's product is again that autologous, and then just we approved just 7 subject data. The clinical data means still they are collecting data to that mode of action kind of data. So yes, that's a conditional and time limited, but that's an approved product. Means already
But they're still collecting, they're still doing
post-marketing phase. So still they are developed the CMC part now. So I think that even the post-market phase, but still like kind of phase 3 situation. So yeah.
So does this bring up any questions from the industry side?
Brings up a bunch of challenges. No, it brings up a bunch of challenges. I mean, the industry's been plagued with this for years. You know, how to define potency for a cell, how to define comparability, to make CMC changes, to understand the safety profile of cells. It's incredibly challenging. I don't think we've got a right answer yet. I think there are some very pragmatic approaches from regulatory agencies, and I think the Japanese situation is one where you know, perhaps I'll put words in your mouth, but it's acknowledged that the only way that we might figure out what sort of the true potency, comparability of cell products is actually to take them into a relevant species, into man, and to follow the progress over time. Now, that will ultimately probably be successful, but it does raise the potential question of safety to the patient, during that process. So I don't think there's a right answer yet, but we will talk probably later about different matrices of assays and things that we can use.
Yeah. Well, that's a good place to go next, I think, is to the matrix of assays. How is that helpful? Francis, you want to take a shot at that?
The matrix of assays helps. It gives you you want to make that link between your CQAs and your CPPs. The more CQAs you can understand of your product, the better. But there isn't really a black and white answer ultimately. I mean, I think with cell therapies, fully understanding the mechanism of action is usually a bar that's hard to attain early in development. And you kind of need a multi-year strategy where you can kind of look ahead and say, these are the changes I'm probably going to need to make to my process. Come up with a strategy based on a risk assessment of how you want to do that. And then I think it's important to reach out to the health authorities and get their opinion, because there isn't necessarily a black and white answer. And so you need to get the perspective of the different health authorities around your strategy and then take that feedback back and kind of work towards that. The more data you can collect and evidence around having a multifactorial assay approach, I think that helps. But there's still always going to be kind of a gray area around process changes and understanding if it's really an impact, if the change can make an impact to your potency or efficacy.
Stephen, what do you think? Francis is bringing you a product. He's got a matrix of assays. It's pretty close.
Sure. So I think matrix approach gives latitude in several ways. So I think one of the interesting ways that the matrix approach can be utilized is that for potency assay, you would ultimately want to have an assay that measures the function bioactivity, and sometimes that is not possible based on the manufacturing scheme, based on your lot release program that you have set up, and the results of that kind of assays might not be readily available, and for timely release of the product. So matrix approach can give you latitude in the way that, let's give an example of a product that you would cryopreserve, and then at the time when it needs to be distributed, you would thaw it, and you would recondition it for, let's say, a couple of days in culture, and that you would send or ship a fresh product. You would definitely need to have another set of release criteria at the end, even though you have done some at the time when you're cryopreserving your product. So potency assay matrix approach can be used in this instance, where you could do your functional assay on the product that you were about to cryopreserve, but have an alternate assay, some kind of analytical assay that you could measure and get results in a short time frame, and have that done when you're releasing the fresh product. And so you have basically two potency assays. The functional assay being done on the cryopreserved product, in that case you would want to demonstrate that that the assay, the function of the fresh product would have the same potency as the one that you have measured on the previous stage where you were cryopreserving. So that's the kind of latitude that matrix approach could also have.
Do the European and Japanese authorities agree with that in lockstep?
Yeah, I always say that the approach is roughly the same. What I would like to add is
roughly is a scary word for those of us in industry approaching.
Now, what I mean is, of course, the matrix assay is a way to compare two products. But if you look at the guidances that we have, the ICH Q5E, for example, the goal of the comparability assay is to demonstrate that the two products are the same. So whatever the test, either it's matrix assay or any other tests, if the two products are truly the same with any assay that you use, then of course there is no issue. You will have two comparable products, and the comparability will show that the changes do not affect the product. The problem usually comes when the assays demonstrate that there is a difference between the two products.
And the difference is a statistical difference, a p-value?
Exactly.
So
exactly.
within a p-value of 0.05 we're okay, outside of it
exactly. What is the extent of the difference? And then, of course, you will have to demonstrate that these differences do not affect the safety and the efficacy of the product. So the way you can compare is one part of the exercise. What is really important is what is the result. What are the results of these comparabilities that you will perform?
Okay, so before we get into the extent of the difference and how the greater clinical development program is affecting what the tolerance level is within that, which we have an interesting discussion about. I'd like to turn to Keith for a moment, who's got a very unique perspective and having been at FDA for a number of years now in industry. And just to address this issue, we talk about process being product and product being process, and just to be careful about that expression, because we know that we're changing processes. And so if all we have is a process, we really don't have a product.
Right.
So we've got to really understand to some degree the product. Could you address the issue, I think Francis brought up before, the CPPs versus CQAs, what that means and how we can use that as a strategy, and then maybe we'll ask a regulatory, because that's an extension of the matrix, if you will, of assays that describe what we're giving to a patient.
Yeah, so I think what we're looking at here is an opportunity to really understand our product. And so the tests that we perform should tell us about the critical quality attributes of the cells. And that, in an ideal world, will tell us about mechanism of action and help us understand mechanism of action, which will help us to understand clinical outcome, right? And we want all of those things to trend in the same direction, right? The test tells us about are appropriate to determine the CQAs, which are appropriate to determine clinical outcome. You know, and if we can get these things all aligning and trending in the same direction, then we start to gain confidence that we understand our product. We understand what it's going to do and why it's going to do it. And so I think if your process is your product, what you're saying is, I don't understand my product, right? I don't understand CQAs. You know, I don't understand my critical quality attributes.
So CQA, we identify, just to be clear, so we're all sharing the same language, is only on the final product? Or can you look at CQAs throughout in the way you're describing them?
So I think that, you know, this is, well, I think ideally it's on the final product, well, because that's what goes into the, because I think our experience at Novartis is we're dealing with autologous patient-specific products, right? And so what is the starting material? It's the cells that we get from the patient themselves. And the starting material is going to vary greatly, right? And so what you're going to do is you're going to take something that varies greatly and you're going to funnel it down to something that's defined, where the CQAs are well defined. Now, are there things that, along the way that tell us we're heading in the right direction? Yes, but I wouldn't call those CQAs. You know, it informs your process parameters. They're in-process controls. You know, and they're important, but I wouldn't call them CQAs because for a patient on this side of somebody on this side of the spectrum, you know, the path here is different than the patient on this side of the spectrum where the path is going this way. So knowing that you're on the right path, the points along the path are different points. So it's not a CQA until it's shared common among them to tell you that this is going to...
So where do the critical process parameters come in?
I think the critical process parameters are telling you that you're on the path, right? That you're going where you want to go. And for us, it's been a great experience to learn, you know, how to adjust to that patient variability. For a lot of the allogeneic products, you know, the starting material is pretty consistent, and so and you can, you know, it's more of a straight line in one line. We're looking at multiple vectors to get to where we want to go, and so, but it's.
So would you have answered that question the same way 2 years ago? Have you seen something on the development side now that's modified that, or was just exactly the same? Because there's no doubt Novartis said Keith knows FDA, knows the regulations, really has a good view of CMC issues, bring them in, help this development. Have you come on the other side and said,
yeah, well, you know, I think one of the things that I've come to appreciate since I've been at Novartis is that there is the potential, and I'm sure all companies are different, but there's the potential to really have a robust understanding and to have a robust process to say, how am I going to, I talked about, you know, having different vectors all shooting at the same target because you're adapting to the variability. Well, that means there's a lot of changes going on to make those adaptations. And there's got to be a robust system to handle that, a robust system that tells you that you can manage change and that you understand when and why you're making the changes and what the risks are and what the reasons are for making those changes. And it's that system, that change control process that I didn't have an appreciation for when I was at the agency, that I see now that there really is a lot more to it than just, you know, does test X equal test X when you have two different products and you call it comparable, you know, but understanding.
Does that modify what you were asked for? What you would ask for if you were sitting in their seat now? Does it change the way, or do you just think that, you know, that was a pretty good thing to ask for? It's just harder than I realized to get there.
Well, you know, I think it's
important that we figured that out.
I think it is important, and I feel like, you know, understanding how much baseline a product developer has and where they're coming from really informs the potential for the comparability protocol to tell you what you think it's going to tell you. You know, and if there's not a robust foundation of science to know what the critical quality attributes are and to know what the critical process parameters are, then you're not going to get the same answer even if your test is within that statistical range that you expected.
So before we go into, I think that statistical range. Oh yes. Yeah.
So kind of want to pick up on a couple of things. So I think the theory around you know, matrix assays is wonderful. You know, identity, purity, potency. I've worked on autologous products, I've worked on allogeneic products, I've worked on gene-modified products, I've worked on non-gene-modified products. Across all of those different products, I've rarely had a problem in defining identity. I've got 300 cluster of differentiation antigens that I can pick from. There are other processes that I can use. I rarely have a problem with identity as part of my matrix. I sometimes have a problem with purity, depending on what level of I set the specification at with regard to impurity. Is one pluripotent stem cell in a billion a problem? Is it one pluripotent stem cell in a million a problem? So purity, depending on the setting, can be an issue. Potency is a bear. And still early in the morning, so I'm going to wake up the audience. Let's do a little bit just audience participation for a second, and I'll try and exemplify what the problem is here. Hands up, everybody who has children. Okay, keep your hand up if you think that your child behaves differently at home than they do in school. Okay, so what we're trying to do with our cell therapies is we're trying to send our children to school. They're going into the body, they're doing something, they're reacting to the environment that they're in, that school playground, And fundamentally, what we're trying to do with our potency assays or all the rest of the functional assays is we're trying to measure them at the breakfast table before they've ever left to go to school. And so we can influence, as parents, we can influence our children, we can influence ourselves in the way that we grow them and the environment that we place them in, and we can measure them. But at the end of the day, we're trying to make predictions as to what they're going to do when they actually get into the patient, into the body, into the schoolyard. And so, you know, when I come back to, you know, I've used, you know, try to assign critical to quality attributes for years, try to assign critical to process parameters for years. But in trying to assign that, we're trying to assign what happens in the schoolyard, what happens in the patient, and making the predictions on what, from where we are, which is either drug substance, intermediate, or drug product, which is getting closer, but we still have a little bit of vagueness with regard to what happens when they leave home. And so, you know, we've tried over the years, and I'm sure many others have as well, to predict that from animal models, and sometimes it's good, sometimes it isn't. But the real challenge, in my mind at least, when we're talking about any of these changes, how to implement changes, how to control changes, how to develop the process over time, how to manufacture at multiple sites, is understanding potency in the body.
Okay, thank you. So before we go on, just a quick note, my children are perfect on both, whether they leave the house or not.
Mine are not.
I don't know how they're behaving. But so are there any questions from the audience around this? We have sort of one more topic we'd like to cover, but I could pick on people. In fact, I'd love to pick on Jiwen, who's done tremendous work analyzing and doing product potency and analytical development work on the Caladrius product. We've been close to that project for a while. And maybe, Jiwen, when you hear all of this, I mean, are there any questions you have either on a global scale or specifically for the panel?
No, I don't think I have actually a question, but I can really relate to the difficulty of having comparability and having a potency assay. And in fact, you know, the current challenge that we are facing is that we have a potency assay in place, and that the regulators have accepted that for, you know, phase 1, phase 2, and they will actually accept it for phase 3, but they have already indicated that they will not accept that potency assay for market authorization. So apparently, you know, they look more stringently, I think, at potency assays during market authorization than during clinical trials. To me, that was a bit of a surprise, because of course now we were faced with quite some time pressure to develop a new potency assay.
That's an interesting point. That'll really come up in Jiwen's part of this discussion then in terms of the accelerated. So I won't get into that right now, but I'd love to ask a question of that right now. But I think that's a great question. So Jiwen, you should pick up on that, I think. Because as we accelerate the development, at least you're in a phase 3 setting, but these other products that are in phase 2 and accelerating, it's even worse, right? It's even more difficult because we know at phase 2 the assays aren't ready. And you know, you have particular knowledge of your product, and you've got quite a sophisticated package, matrix of assays that describe what these cells do.
But like you said, I mean, the difficulty, of course, is that all you have is a proposed mechanism of action. And you know, if you have a mechanism of action, then, you know, for us, for instance, what we need to show is we give some cells to a patient. They don't need to cause GVH. For that, we pretty much have a good assay. What we would like the cells to do is to challenge, you know, we would like the cells to fight infections and all kinds of pathogens. Well, there we could find a pretty good assay as well. I would like the cells to immune reconstitute the patient.
Right.
I don't have a clue how to develop any potency assay for that. So, you know.
Good. Well, thank you. You've got great experience. If anybody has any questions about this sort of thing, I think Jiwen's a great guy to reach out to if you're willing to answer them. So I just have one more quick question to go through the panel. We've only got a couple of minutes left, and then we'll turn it over to the second part of the session. When we talk about these changes, is the reason for the change important? Does it matter why we're making this change? And one way to look at that is if we're making the product better, if we're making it more potent, does that have any difference in the level of tolerance you would have and the way we would look at this? I'd like to get to the point talking about pre-launch and post-launch, but we're not going to have time. But just in terms of the framing, maybe we can just have a quick answer. We've got a couple of minutes, and we'll start and just get a little bit of a thought from each of the panelists, and then I'll move on.
No, I think this is a very important issue because the reason for the change is very important for the regulator. Because as you say, it is just to increase the yield of the cells, to increase how many cells are produced. Of course, these changes should not really impact the potency of the cells, because what you need is just to increase the amount of active substance or final product that you have. Instead of modifying manufacturing process to increase the potency of the product itself. So, for example, we discussed this yesterday, for example, for the CAR T cells, if you want to increase the viral copy number of the cells, just to increase the efficiency of the cells, this, of course, will be considered as a kind of major change because you are really intended to modify the potency of the cells at the end of the process. So, of course, I think you're right. In the discussion that you will have with the regulators, it is important to justify the change and the reason of the change that is under the modification.
And critical, which will come into that next discussion, then is post-launch. These changes are much more difficult in general, regardless of the reason. So, Stephen and Yoshiaki, I think you agreed with that approach yesterday, but specifically, if you don't agree, please say so. I don't put words in your mouth, but I wanted to move the question a little bit to you as we move down the line. Is when FDA looks at the change, if we're making a change because we have a way to improve on the sterility assurance of the process, versus we have a way that will make this $50,000 cheaper. Do you look at that change any different? It's not a change in potency. The reason we're making the change is for safety reasons versus for cost reasons. Does that impact the way you look at the change and evaluate it?
I think we would look at in subjective way, but we would also look at how that would impact the actual product, the quality of the product. If you're making an improvement you're making a change to introduce a different reagent, for example, which for a reason that it is no longer available, or for a reason that this reagent would bring down the cost of the manufacturing costs. I think that is important, but not as important as are you really making any changes to the product by doing that, by making that change. So we look at it from that perspective as well. I do agree with Nicholas that if you're trying to improve the potency of the product, okay, that's great. But where are you at in your product, in your clinical development? Are you just starting out your phase 1 study and trying to change the potency? Maybe that's okay, because we're still looking at the safety aspect of that drug through a clinical trial, and the efficacy will come afterwards. But if you're trying to do that in the middle of phase 3, okay, what about all the clinical trial data that you have on a product that was less potent? How would that be applied to support your marketing application, for example? So that kind of thinking needs to go in. I don't think there is a straightforward answer to that, but the scale of how you would apply that kind of thinking, I think would go up as the product development, the clinical phase of the clinical development is going towards
really
later.
This risk-based approach to
that
evaluating.
is exactly right.
Yoshiaki, from your perspective, the post-market approval versus the conditional approval phase difference. I mean, there was an interesting perspective you had on this. So.
Actually, yeah, basically same. We assess the impact for the effectiveness and then safety. So that's basically same.
Very basic.
Yeah, but yeah, and but that phase is definitely different the knowledge because they are the CMC is a developing phase. So if the post-marketing phase, they collecting lots of data to identify your product, then that I think that the comparability tests for these assay also can be do. So I mean that so the post-marketing phase still lots of information compared to the
yeah, we're getting a lot of information.
Yeah.
It'd be a shame not to be able to use it.
It's a different.
Yeah.
And then, actually, I mentioned you mentioned talk about the sterility issues.
Mm-hmm.
If use sterility test for the pharmacopoeias, it takes 14 days, right? So if the sponsor wants to change to the rapid microbial method, yeah, that we are very encouraged to this method. Because,
in my discussions with you in Japan, you didn't encourage. Well, you did.
Very
It was very encouraging. You helped us develop a rapid assay.
Yeah. Because that, yeah, you know that.
No, yeah. It's fascinating.
Yeah, so that it's still kind of comparability, but if the assurance or the safety issue, then it's not so different, means we are acceptable to changing the method.
Right, thank you very much. So we're unfortunately out of time for this session, but now we'll move on to the better part of the session. Dr. Jiwen Zhang is the Senior Director of Regulatory Affairs and Cell Therapy and Regenerative Medicine Group at GE Healthcare. We'll talk now about how accelerated approval and how the clinical groups are getting a little out ahead of our ability. We can't really catch up, and then following that, Mike Mendocino will provide a summary of the meeting. Thank you very much.
Thanks, Bob. Actually, we did. We had a great preparation discussions for the last 3 times, and we're hoping some of those will come out here as well. But we'll make it as free flow as possible. But I do want to pick it up from here. So, a couple of things, right? We talk about the process development and process changes. I think what we also want to, Bob talked about the survey, what are the challenges are, so we'll skip all of those. But let's focus on, we do have the pressure to accelerate because like the conditional approval, you got the products on the market, now you really have to keep up with the supply and also make sure you understand the products quite thoroughly. So can we, whoops, what did I do? Can we truly accelerate CMC development from one is to align with the clinical development, but the other one is just really to try to align with the overall program, especially if for some companies they may have multiple indications for this particular product. So the other aspect, actually, the second block of the questions we're trying to, as a Alliance Regenerative Medicine Science and Technology Committee, we've been trying to really get stakeholders together to see how we can work together as an industry, but also importantly work with the regulators to identify ways, either mechanism-wise or some regulatory mechanism-wise or some kind of industry contribute on data-wise that we can truly help move this field further ahead through accelerated CMC development. So those are really the two kind of topics we're trying to get through a little bit. We would like to have the audience to contribute your thoughts, especially how you can try to, how you have been trying to accelerate. But let's start with the panels, because I think we had from our prep sessions, we had a lot of good approaches, and actually Mike Kelly was here, talk about some platform approaches yesterday. I thought was very interesting as well. But let's start with maybe the sponsor side. Keith, Stuart, from normally from your company, how you try to accelerate CMC development?
Yeah, so our experience has been that a lot has gone into the understanding of the product early so that we can facilitate the later stage development. And I think one of the things that has been impressive to me is the breadth of knowledge that has been gained on our product and in terms of the different analytical approaches that we've taken to characterizing the product. I think one of the other things that we've done that has been very helpful is we have a frozen product, and Novartis has been very diligent in saving. It's autologous, so it's patient specific, and we have saved aliquots of all of the batches that we've created, and so when we develop new methods and new changes, we can go back and we can look across all of the patients and reassess with the newer technologies, and that has greatly helped us in many ways to accelerate the development. And I think one of the other things that we have done well is we have used the development group to really explore lots of different options in parallel. Mm-hmm. And so there hasn't just been one effort to say we're going to do things this way. It's been we're going to look at 3 different things in parallel so that we can then be in a position to implement the most effective approach. And so those have been some of the things that we've done that really have helped us from a CMC side to accelerate the CMC development.
Fran, you want to?
I guess coming from big pharma, where you see a large portfolio of products, and you kind of see that maybe only 10 or 15% of products actually make it. So it's not like you're a small biotech and you have one product. So you look at that and you kind of realize the ones that make it are the lucky ones, and getting that understanding early as possible to try and push the product into the winning category as opposed to all those ones that are going to fall out is really key. So I agree with Keith that early understanding, early analytics is really important. And it's very hard to accelerate that because it's difficult, time-consuming. Developing potency methods in animal models is difficult, but really time-consuming, but worth it in the end. I think in terms of acceleration, looking at your commercial strategy, especially I think with big pharma where you've commercialized lots of other products, you try and imagine what the commercial strategy, what the commercial process and product will look like. And so you try and incorporate a lot of those aspects into earlier development and not to leave them till phase 3 or post-approval. So incorporating things early, even if it slows things down a little bit earlier on, that certainly helps. I think another area where I'd like to see improvement over the next few years is around the platform technology, around the unit operations, working with vendors, suppliers, because I think each company is really going it alone and trying to develop their own technology. And if we can kind of work together and have off-the-shelf operations that are very well understood, that we already have understanding going into those unit operations, I think that will really help us accelerate moving forward. And I kind of see us building up some head of steam in that area. So we're not quite there yet, but 5-10 years down the road, I think it'll be a lot more plug and play.
Stuart, you have both, right? Large and small.
I do, and it's an interesting set of trade-offs. So, I mean, I agree with what's been said already. I mean, the more you can plan ahead, the better. I've been in larger biotechnology groups, pharma groups, who have had the luxury of spending 2-3 years on process development before ever thinking about taking a product into first-in-man. And it is a luxury. I'm sitting now in a small biotech, having moved a product from concept to first-in-man in less than a year. We know we've made trade-offs. We know that in moving into man as quickly as we have, there will be process challenges down the line. But to the point that perhaps only 10% of these candidates actually make it, and it's difficult to predict that from the animal models, we chose to move into man with what we believe is a safe product, a reasonably well-characterized product, a reasonable process that can be scaled, but knowing that it will have to be changed down the line should this be shown to have safety and efficacy. Whereas previously I would have spent a lot more time up front assuming that it was going to succeed and putting all the process parameters in place such that we didn't have to make too many changes down the line.
So if I summarize a little bit before I ask the regulatory experts to answer, I think we touched on a couple of things, right? Including yesterday, we touched on how about using QMS approach if we were able to accommodate or really justify the process changes without getting too much of adequate regulators stamping on it. That's one thought. The other thought we kind of came out was about this platform approach, including collaborations, etc. And the last one I think talk about was several pathways, right? Potentially you develop different options, alternatives, and then either take one or multiple to the regulators to get some feedback. So from regulator side, pick anything you think that has been successful, what you see reviewing the products.
So I can go first.
Yes, please. Steven.
So in terms of keeping with the accelerated CMC development with the rest of the product development, the clinical development, and Keith mentioned that eloquently, that you would really want to, at least from our point of view, that try really understand what your critical process parameters are and what your critical quality attributes are. If you are in the early stage and still trying to figure out what the quality attributes are, let alone what the critical quality attributes are, that's still on the right track, I think. You would want to explore as early as possible to try to really understand what your product is, what your manufacturing is doing to your product. If you don't have that information, and when the clinical development is getting ahead of CMC, there's a catch-up that needs to be done at some point. And that, from our perspective, that process has been very difficult on the industry side. And we see a lot of those activities going on, and that has not been playing out well. So I think our advice would be that try to engage into launch a CMC program that really looks into your attributes, manufacturing process attributes and product quality attributes, and try to understand what the relationship between the critical product process parameters and the CQAs are, and then you use your classical risk approach, and this is the concept that's already been out there. It's an ICH concept, and you try to prioritize what is important, what risk is important to you in relationship to your CQA, and therefore this change that I'm about to make, how would that impact in terms of that risk that I have just evaluated. And that sort of drives how you would approach your comparability study. And I think having that kind of basic information about your product is the essential part of all this in our view.
Thank you.
Now, I fully agree with Stephen, of course, but what I think is that I'm not always very comfortable with accelerated CMC development, because sometimes it's better not to go too fast. If you need to accelerate, that means that you know that the way is long to get to the final goal, meaning that probably you are not satisfied at present of your manufacturing process. Meaning, of course, that you want to improve it. There are some uncertainties related to this manufacturing process. And I think that regulators usually don't like uncertainties. So the problem is maybe for you is what is the delay to reach a robust manufacturing process? And then, of course, before you have reached this very robust manufacturing process, which is, of course, the best way to get to a marketing authorization for the regulatory authorities, the timeline between where you are now and what is your goal, of course, you want, I mean, as a sponsor, I think you need to reduce this time. But at the same time, I would say, don't be too fast to improve this process, because if you don't know, and once again, I agree with Stephen, it is better to do that before, of course, before marketing authorization, but even sometimes before getting to the patient, because once again, if you have to modify, to have major changes in your manufacturing process, this will probably also mean that you will have to redo some preclinical and clinical assays. So it is really difficult, except to reason on a case-by-case basis, to have a kind of overall framework for what is accelerated development for cell therapy product. I would say, except in a very specific situation when you have a clear medical need that is really unmet. And then, of course, you have patients, for example, that are really willing to have a product available for the patient. In that case, maybe it's the only case for which the regulatory authorities will really understand what is the accelerated CMC development for a product. If you don't have such a pressure from the patient, usually I would say, don't go too fast. Just be sure what you are doing and in which way you want to improve.
I'm going to challenge that, but before that, I want to hear, yes, Yaki-chan, your...
I guess I don't have no more comments, but it's a kind of same approach, we think so. But yeah, there is lots of options for the quality systems. And yes, we have the experience for the medical device company applied to the product. So that means they use the QMS approach, right? But it's a system is name is different, but basically the same approaches. Mm-hmm. You said the critical process parameter and the CQA is important. They also, and they understand that. So yeah, basically it's the same.
Thanks. I think what I was going to challenge is I remember when I was doing when I was in biopharma doing clinical program development. My clinical directors, when we design clinical studies, wants to know everything. And my statistician would say, no. This is the first primary endpoint, secondary endpoint, and also, right? You have to sacrifice. So I think CMC is the same thing. We would love to know everything, know as much as we can, but unfortunately, business pressure, clinical development pressure, or patient pressure, we do have to prioritize and try to identify ways to be able to somehow, maybe accelerate is a little bit of a sensitive word, but somehow to find an approach that we can really satisfy regulators, but also importantly to be able to make the quality product. So I think that's the spirit or intent here. Trying to see if we can find some a best approach. Yes, please.
But if we don't accelerate, then we have to make changes post.
That's true too. Yes.
Hi, I'm actually an ex-colleague of Nick. I'm a member of the MHRA, and I also sit at CAT. And I just noticed that I think when you said, I normally agree with most things you say, but on this occasion, I think it's not been made clear enough that getting the accelerated approval will put you in a situation where you just basically have to scale up or supply to more patients. So it's not just a question of, having not got a good enough process, but maybe having a challenge with suddenly having a bigger patient basis. And I guess I was interested to hear the commercial, the industry's perspective on that. And obviously, one of the things to do is throw money at the problem. But I would personally always think that good knowledge of the mechanism of action and the potency, which is what Nick said earlier in the first session, is always the key to be able then to make changes to the more easily you can basically separate your background noise from from what you actually want to see, then the easier you're going to find it to make your changes.
Yeah, so I can start by saying, one of the things that, I don't know if it was clear in how I pointed this out, but one of the things that we have found has helped us as we accelerate our development is making the right assumptions up front, right? So, for example, I said we kept sample of all of our product that we released so that we could go back and do retrospective testing. Because the assumption was there that the process would change and the test methods would change. Mm-hmm. And if you have no sample to do look back, then you can't support those changes. You have to repeat the change. You have to repeat all that data, or get all new patients. And so there are some things that you can do if your assumptions are that you know your clinical results are stellar, and that you're on an accelerated path, and that that you're going to be adapting your process, you're going to be adapting your methods, you're going to be developing that new potency assay to get to commercial, and if you don't have opportunities to look back and to do that, and you're not doing things in parallel, then it really impedes your ability to be accelerated. It actually just slows you down because you're repeating yourself. But at the same time, I also think that there is a reality that things take time, like Nicholas said, and there's throwing more money at it is one solution, right? Lots more people and lots more money, but it's a luxury that not, like Stuart said, it's a luxury, right? That some people have, and so, but if you see your program going in a certain direction, it's planning and knowing that is what's going to have to happen.
So, Jiwen?
Yes, please.
Sorry. So I'd say, one observation, this might sound silly, but it's actually relatively easy to develop, to accelerate CMC, as long as you can define your critical to quality attributes for your product or your process. But it's really difficult to do that. And I'll give you just one example. Several years ago, working on a mesenchymal stromal cell product, done a whole genome screen with regard to defining what we thought were 2 critical genes related to mechanism of action based on our in vitro assessments. And the in vitro assessments were actually pretty robust. I won't go into all of the details, but we thought the in vitro assessments, functional assays, were quite robust. We locked it down on 2 genes of interest that we thought were critical to the quality of that cell product. And therefore, if our process changed those genes, then it would be problematic. And if it didn't change, then it would be fine. I then suggested a really dumb experiment in hindsight. Not dumb because it was a dumb experiment, it's just dumb because it didn't work. We took the two genes and we knocked them out one at a time in the cell lines. And we took them into what we thought was a predictive animal model. And I'll give you the animal model just so, and it was a EAE model of multiple sclerosis, or just basically inflammation. Pretty robust model, works, it's got a good dynamic range, got good sensitivity, can work in a number of different ways. The animals tend to show the same results. Took the cells with this critical to quality attribute knocked out into the animal models, and there was no difference. There was absolutely no difference. And it's beautiful in the in vitro assays. Took them into animals, didn't make a blind bit difference. So we spent 6 months trying to get to a critical to quality attribute such that we could change our process, and ultimately didn't have a critical to quality attribute, and therefore didn't know how to change the process at that particular time. And so it comes down to how do you define those CQAs? It's tough.
I think it's a learning experience, right? And actually, as you go through your clinical program, you understand more about your clinical data. That also helps you on the critical CQA side as well. We have actually, that goes really fast. Before yes, please.
Agree that the sooner you start characterizing your product and your CQAs, and preferably I would do 3 or 4 years of CMC manufacturing development before even going into man. But reality is, no way. I mean, I work for Caladrius Pharma. We're a small biotech company. We're public since a year, so we have a little more money on our hands, but far from unlimited funds. And before that, we were owned by venture capital. Reality is, we bought this academic process. That was the first phase 1. And then, of course, timelines are very limited. Budget is very limited. I need to improve on the manufacturing, but I need to be ready for phase 2. And so consequence is, yes, I am going to make changes to the manufacturing process throughout the whole clinical development. And of course, it's going to be based on the risk assessment. Yes, I try to, identify what my critical quality attributes are, but I have to develop manufacturing because I cannot take that academic manufacturing process into anything resembling marketing, a commercial process. So I don't really have a solution for it, but it obviously means that the product that I manufacture now and the product 5 years ago is going to be different. For instance, in terms of it's going to have a better purity. It may even have a better potency, and I don't really know how to deal with that exactly, because does it impact really the safety of that very first phase 1 trial or not? What is the risk associated with it? I don't know if anybody has any guidances on that, but simply the let's say the perfect solution of first really developing that whole manufacturing, characterizing the product, and then going into man, I think for most companies just is not an option. It is going to change, and so.
Thank you. Yeah, actually, with that, that's why we put the next block on the discussion panel. Is really with individual companies, it's difficult, and as ARM collectively trying to bring the stakeholders together, can we really work together, especially with the regulators? So now I'm putting the regulators on the spot. So if we do, as an industry, work together to bring something to you, what would you like to see that can help to move forward, help with this field? Stephen, sure, please.
Sure. That's a good question. So I'll speak for FDA. So FDA, as you all know, has its own limitations, which are bound by the laws and regulations. And there are things that we could discuss openly with people other than the relationship between the FDA and a particular sponsor. But there are many things that we're not able to discuss. So it brings it raises another topic as to sure you can as a group collectively raise issues and try to discuss with FDA, but how that is going to happen is not clear to me. Doesn't mean that we're not open to that kind of interaction, but at this moment, maybe there is a creative way to do that, but at this moment it is not clear to me. So I'll leave it at that.
Thank you. Nicholas?
Well, I can't speak only for France, because as long as you are in the clinical development of the product, the clinical trial authorization remains in the remit of each member state in Europe. So it's difficult to have a European perspective on this, and I think that maybe Christian may have a different position than I, because she's in the UK, I'm in France. But I would say that, globally speaking, I agree with Stephen, that of course, most of the time, when we are dealing with small biotechs, we have to reason on a case-by-case basis. Because for us, I mean, I agree with you that when you are performing a first-in-human clinical trial with a product that has been manufactured by academics, which should not be the case in Europe, because we need to have GMP products at the very beginning of the clinical development. But anyway, it may happen in some member states. So the question for us is, what is the normal pace for the development of the product? Meaning that if you go to the patient, normally you have to wait until the results of the first trial to modify the product, and then to come back to the patient with a, I would say, a modified product in a second phase of development. So it really depends on the indication of the product, because obviously if you are dealing with patients in oncology, for example, patient who will have a very short life expectancy, you will really know very rapidly if the product is efficient or not. Let's say for the CAR T cells, now we know that it is very efficient, but in the very beginning of the first versions of the CAR T cell, it was not clear whether the product was efficient or not. So if you may have clinical endpoints that are very strong very rapidly, then of course you may in a certain way accelerate the development of the product. But if you have to wait for 2 or 3 years to know exactly what is the outcome of the treatment, I don't see how you can deal with CMC development together with the time needed to get the results of the clinical trial. Of course, this is if you want to modify in a certain way the potency of the product. If the modification are only, I don't know, are only dealing with safety, of course, then you may rely on preclinical studies to demonstrate the comparability. And once again, we are back to the ICH and the comparability exercise that we know. So I would say that what is important is it's really a case-by-case analysis for small biotechs. And I would say also what is really important for us, and I think for you too, is really the risk analysis of what you are doing and how this will impact the product before acceleration.
So you mentioned case-by-case quite a few times, but I have a question for you, which is, you know, in terms of getting more concrete guidelines, do we have enough guidelines in cell and gene therapy?
No, that's a joke. I just, during the preparation of the meeting,
come on now. I set him up. He said it so well yesterday. And
I think this is the true situation. In Europe, we have more guidelines, guidances, regulation, and directive than we have product on the market. So I think that my perspective that we have enough guidelines, because in each guideline now it is written the evaluation will be on a case-by-case basis. So, but I think it's really difficult to get out of this, because I mean each company now, each cell, each indication is different. So I think overall it's really difficult to have.
Yeah. So along those same lines, if I could ask one more question. So you know, I think for many years in the industry we've know that cell and gene therapy has been excluded from ICH guidelines, and so I feel like we've almost ignored them, right? Can you talk about the application of the principles in ICH to cell and gene therapy?
Just a few words. If you look at ICH S6 for the safety guideline, it is clearly written that this guideline is for biological, with the exception of cell and gene therapy. And if you look at the guideline, the cell and gene therapy European guideline, it refers to ICH S6. So it's difficult to know, but I would say that it's a good basis, at least for the beginning of a comparability exercise.
Just to let you know, we had, 3 preparation, 4 hours discussion, and we have more to discuss. So anybody, after the meeting, please, feel free to come forward. I was going to ask Yasuaki-san about Japan's perspective. Can you just a couple of sentences before I move on to Mike?
Okay.
Or either way,
maybe
fine.
just ask the
Please,
question.
then. Basically, we were trying to say, hey, with industry together, we would like to work with the regulators. What would you like to see we bring forward to you from PMDA's perspective? Can we bring some data to say, hey, RCR requirements are a little bit too much? Can we simplify it? Would that be something you would consider from FDA, PMDA or PMDA now?
I'd like to say case by case basis. So I mean that that we have a R&D strategic scientific consultation. That option is very frankly communicate that problem to sponsor and PMDA. So yes, if you have get some challenges on your product, please contact us. We are happy to share that information and then solve your challenges.
And I would just echo that from the FDA side as well. You would want to engage us in a discussion as early as possible so that you will have a tailored discussion and guidance for your particular case. In doing that, I would also emphasize that prepare well your briefing documents, prepare for all 3 disciplines, which are the CMC, preclinical, based on animal models, and all the studies that goes with that, and the clinical portion of it, and ask thoughtful questions. And when we put a lot of effort into providing responses, very detailed responses, as long as the quality of the briefing package is there. So I think that's very important that you prepare for your briefing package.
Thank you very much. It's been a great session. As you can tell, there's a lot of interesting topics, and I think one of the outputs of this is really going to be we need a pre-conference half-a-day or full-day workshop to really fully dive in. So just a couple of additional comments before we close. So it really is important to get all the thought leaders together from industry, regulators, former regulators, all bringing their perspectives in for this type of collaborative conversation. As Jiwen pointed out, we did have some pre-session discussions that were also very lively, and we're hoping to capture all of this, both from the Science and Technology Committee perspective at ARM, but also for the Regulatory Committee, for all of us to work together in order to perhaps have some outputs and some projects and initiatives to work together, both from the scientific and industry community perspective, but also from finding ways to work with the regulators as we determine what those mechanisms will be. So there are differences, say, for example, with regards to GMP requirements throughout clinical development in the different geographic regions. But there are some common denominators in the ICH requirements, and although maybe more applicable to allogeneic off the shelf, that there are definitely, as Keith pointed out, existing and relevant ICH guidelines. But it was also mentioned, jokingly but true, that if there is truly is more guidance than there is approved products, we don't want to make our situation more complicated than it already is. So hopefully there'll be some more simplistic methodologies taken towards this approach. If we talk about the requirement for early product characterization and how that can benefit one when you're looking at manufacturing process changes later, one way to accelerate CMC development is just because you're not required to have a potency assay until phase 3 doesn't mean that you can't do more earlier on in order to accelerate your own CMC development, not necessarily wait for the regulators to tell you how to do it. Another way to look at it is if you're accelerating clinical development and there's for example, accelerated approval and requires a phase 4 clinical trial, well, if the accelerated CMC development, there's going to be an expectation that more data is going to come post-approval, as our Japanese regulatory colleague had mentioned. One way that the industry and regulators can work together is in the realm of standards. So there's a new initiative standards coordinating body. There is existing standards development organizations such as ISO and ASTMI where the regulators are permitted to engage, and that is one type of forum where these types of discussions and interactions can potentially take place. So with that, I think we look forward to a future workshop on this topic that we can hopefully not only determine more about what the questions are, but also get to some answers. Thanks, everyone. Thanks for the panel discussion, and enjoy the rest of the day.